The Punnett square. How did the recessive characteristic reappear in the F2 generation anyway? Devised by the British geneticist Reginald Punnett, the Punnett square is a visual technique that helps analyze the results of crosses. Let's consider the cross-fertilization of the P generation, a true breeding round-seeded plant with a true breeding wrinkle-seeded plant. It starts out as a box divided into 4 squares. All the different genotypes of the gametes from the round-seeded plant is written along one side, one gamete genotype above each square. All the different genotypes of the gametes from the wrinkled-seeded plant is written along an adjacent side, one gamete genotype next to each square. In each square are written the allele above it and beside it. By convention, we write the dominant allele first. In each square, written the allele above it and beside it. And the genotype for the F1 generation are all heterozygous. Since the dominant allele appears in all offspring, it explains why the F1 generation are all round-seeded. Now we'll self-pollinate the F1 offspring. The different alleles of the gametes of one parent are arranged along one side, and the different alleles of the gametes of the other parent along the adjacent side. As before, the alleles above and beside the square are written inside it. This one is homozygous. Since homozygous could also refer to 2 recessive alleles, we differentiate the two by saying that this genotype is homozygous dominant. This is heterozygous, where the dominant allele from one parent plant contributes to this genotype. Whereas for this genotype, the dominant allele is from the other parent. Even though both genotypes look the same because of the convention in which we write them, it is important to note that the heterozygous genotype appears twice because mom's dominant allele and dad's recessive allele appears in one of the genotypes, and mom's recessive allele and dad's dominant allele appears in the other heterozygous genotype. And finally, the other homozygous form, homozygous recessive. When the genotypes are translated into phenotypes, we can see how the 3:1 ratio of round to wrinkled came to be. You should practice as many monohybrid cross questions as possible. Your textbook lists many. Probability and inheritance. Probability is the study of outcomes of events. Flipping a coin reveals one of 2 outcomes. Dividing the 2 numbers, you get 0.5. The probability a coin flip will reveal heads. A roll of a dice reveals one of 6 outcomes. The probability of rolling a 4 is 1/6. The McGraw-Hill text doesn't seem to have anything on probability. They did try to make up for it by adding in the handouts, and I've made them available for you. I can guarantee there will be questions on probability in my exams. Ideally, probability is represented as a frequency, a number expressed between 0 and 1. It can also be written as a percentage or fraction. For numeric response questions, read how the probability answer should be expressed carefully. Shame to do all the work, find the correct probability, but express it incorrectly. Again, practice is going to pay off. Do the questions in the handouts that I've made available for you. A test cross is used to determine the genotype of a dominant phenotype. Since the heterozygous genotype has the same phenotype as a homozygous dominant, it's difficult to tell them apart. Black sheep have coarser wool than white sheep. It is preferable to have white sheep. If a farmer is trying to buy a white ram for breeding purposes, he wants to make sure the ram is true breeding white so that no black sheep offspring come about when the ram is paired with a heterozygous white sheep. Pairing the white ram with dubious pedigree with a black sheep will ensure all the offspring are still white if the ram is a true breeding white. Incomplete dominance is where the heterozygous genotype produces a blended phenotype. A red flower pollinated a white flower, a pink flower is going to result. Neither allele at the gene displays dominance. No dominance since a third phenotype evolves from the heterozygous genotype. A new allele nomenclature needs to be formed. Pick the letter that matches one of the homozygous phenotypes and use that letter to identify the gene. So here, gene R has two alleles, R1 and R2. The heterozygous cross reveals all three color possibilities. Pay attention to a key if one is present, and be aware what could constitute a blend. It's not always pointed out. Codominance is where both alleles are equally dominant. Again, neither allele is dominant, and so while the heterozygous genotype might appear a blended phenotype, closer inspection reveals both alleles are expressed. An example of this is blood type. This was covered in Biology 20, but it might be useful to review it again. Multiple alleles. With blood types, you notice there are actually three alleles that can occupy the gene for blood type antigen. The A allele, and for the A antigen, the B allele for the B antigen, and the O allele for neither antigen. Considering co- or incomplete dominance, you have a maximum of three phenotypes using two alleles. One phenotype is when one allele is in its homozygous form. Another phenotype is when the other allele is in its homozygous form. And the phenotype derived from the heterozygous combination of the alleles. However, it is known that a single gene is responsible for the 22 patterns expressed in clover leaves. As this image shows, there are multiple alleles, 7 to be exact. If you were to do a test cross on any of these rabbits, the test cross would be against any of the phenotypes with the albino. Multiple alleles are frequently part of a dominance hierarchy. In this case, the wild-type condition that is the most dominant phenotype is red eyes for the Drosophila melanogaster, or fruit fly. Non-wild-type phenotypes are due to mutation, a subject we'll get into later in this course. If the genotype contains the E1 allele, it doesn't matter what the other allele is. The phenotype is wild-type. If the genotype contains an E2, but not an E1, the phenotype is apricot. The most recessive is the E4 allele. Again, test crosses would be with the white phenotype. So genes that have more than two alleles is said to have multiple alleles. There are still two alleles for any given gene in a diploid organism, but the alleles could be two from a range of many more.